Okay, so let's now move to transcriptomics, which is, again, looking at the study of transcripts or RNA. So here we're studying all of the RNA molecules. That includes mRNA, messenger RNA, ribosomal RNA, transfer RNA, and other non-coding RNAs. There's actually lots of types of RNAs. The main one that people focus on is mRNA. Oops, remember, messenger RNA is telling you, it's the one sending the message, right? It is the one that is telling the body what proteins to make. It's the messenger that came from DNA to make proteins. So if you were to do a transcriptomics study, you could collect RNA from either an entire organism. This would be a little hard for example, if I wanted to get all of the RNA out of a human, that would be hard because I literally would have to extract every single piece of RNA out of one person, right? I'm not going to like squeeze out or crush up a person and get all of their RNA. So usually we're looking at the RNA in a particular tissue. So you might have a sample of a tissue or a biofluid. I might just be interested in what is the RNA levels that are in the blood, for example, that are circulating through an organism. You could get all of the RNA out of a small organism. For example, if you're interested in the transcriptomics of just a cell, and you had a cell population, you could then extract all the RNA out of those cells. You could do that for something like a cell culture, if you were looking at cancer cells or liver cells or anything like that. Or you could extract it out of a whole cell organism, like a bacteria cell, if you're interested in what was going on in a bacteria. But when we're talking about large organisms, generally, because the RNA levels are different all over an organism, right? The messages that my liver are sending are different than the messages that my brain are sending, and they're different than the messages that are circulating in my blood. So usually we're just focusing on one of those areas, a tissue or a biofluid, and getting information about the RNA there. Now, transcriptomics, something that's nice about it that is different from genomics is it's going to give you a snapshot in time of all of the transcripts in a cell. So it's dynamic, right? It's giving you a whole account of all of the processes that are active or dormant at that time, right? Remember, your DNA is just the main blueprint of your body. We don't know which genes are turned on or turned off on that DNA segment in a cell. Your liver cell might be upregulating a particular gene, while another cell in your heart is not upregulating that gene. So transcriptomics can tell you this. If you're looking at a particular area, you can get the gene expression, you can get gene expression information from the RNA by sampling your tissue or biofluid. Now, I want to be clear here that although you are measuring the RNA, for example, the messenger RNA, that doesn't necessarily correlate to how much protein is ultimately made or is currently made. So just because you have sent out a bunch of messenger RNA in your heart to upregulate a particular protein, it doesn't necessarily mean that all of that protein is then going to be made. This is something that we actually know now. We used to try to correlate them. We used to say, oh, however much messenger RNA is there, that's how much protein you're going to make. So they're connected. While generally they're related, they don't always correlate. And I want to just be careful to make that clear here. Again, transcriptomics is useful if you're doing a comparison analysis. For example, healthy cells versus some disease cells or cancer cells or any type of pathological cells, maybe cells that are treated with a particular toxic substance. So you're comparing two things together. And you're going to see how the gene expression is altered between those cells. So usually you will collect all of the RNA and then you tag it differently depending on which cell it's coming from, so you know which cell it's coming from with a fluorescent probe. One of the main ways you can measure transcriptomics is by doing something called a microarray. I kind of showed you a little bit of what a microarray is here. So if you collected all of your RNA segments and then you tagged them, what you can do is take a chip that has a complementary RNA segment on each well. And what you do is you put all of your sample into each well and you see if any RNA binds. If it binds to the complementary segment, it will light up. So what this tells you when you read this chip is, you know, for example, which RNA segment was in this well. And you can see, for example, here that neither sample bound. So neither sample had RNA that was transcribed for this particular gene, this RNA segment. This next well over, what you can see is it is a red color. So that means that only the pathological cell or the disease cell in this case, which is a red probe, expresses this RNA. Now, if it's present in both, for example, if they both equally express it, then it would show up as yellow. And these chips are huge, and you do a vast amount of them because every well has to have a different RNA segment in it to see if it binds to that. And you just go through all through the chip and see what genes are expressed and in what level, and you can get an idea of the type of RNA expression that is happening in your disease versus your control. Now, another type of technology I'll talk a little bit about is called Rna-Seq. And this one is accomplished by essentially reverse transcribing RNA back into DNA and then sequencing it. And it does this all very fast, so it's going to sequence each of the codes. And you would use something similar to like the sequencing we talked about for DNA to do this. So the first thing you have to do is make it into short segments of DNA. So you take your RNA, you reverse it back into DNA, and then you sequence that DNA. These two areas are very hot right now, Rna-Seq and microarray. Rna-Seq is the newest area of technology in transcriptomics. And I just wanted to give a little bit of comparison between the two of them because microarray and Rna-Seq both have some pros and cons when you compare doing one or the other, why you would do one or the other. For example, if you look at how fast they occur, microarray are much faster. It takes 1 to 2 days per experiment to get all of the data from a microarray versus an Rna-Seq, which can take up to a week to do all the full sequencing. If you looked at the amount of sample you need, Rna-Seq wins out because you only need up to a nanogram of RNA from your sample versus a microgram for microRNA. Rna-Seq is much more labor intensive because of the preparation and data analysis you have to do to read all of the sequences, whereas microarray is pretty low labor intensity. As you can see, you literally tag things and put them on a chip. The chip's pretty easy to read because it's just color coded, whereas Rna-Seq, you have to actually get all the information from the sequence. Now, if you were to do an Rna-Seq versus a microarray experiment, you don't really need to know anything going into an RNA sequencing experiment because you're sequencing everything. It's just going to give you all of the sequences out. You don't really need to know if they're useful or not. You can determine that later. When you do a microarray, one of the things about them is that they're limited to the chip size. You can imagine that having one of these chips that literally has millions of RNA probes on it would be very hard to make. So usually people limit the chips. They might only have a couple hundred or a couple thousand RNA sequences on a chip, and they'll kind of cater what RNA sequences are attached to the chip based on what they think might already be there. So, for example, I'm not going to put RNA sequences that I know come from a banana if I'm analyzing a set of RNA from a human, right? Or even if I was going to look at some RNA from a lung cell, I might not include RNA from brain or other tissues that I know are only in those tissues on my microarray chip, because why bother? It's just going to be a bunch of empty things. So you kind of have to know a little bit about what you're looking at before you do a microarray versus an RNA-seq. Now, conversely, the thing about RNA-seq that is good is if there's something there that we don't already know about, then it will find it because it's going to sequence everything. Now, then there's just some differences in how sensitive and the range that they have. RNA-seq usually also wins out in this sense because the sequencing is going to be more sensitive than in a microarray.